Hey guys, Mrs. Shelton here and
Mr. Acorn.
And we're here to introduce to you Activity 5 Gene Squares. So before we begin, let's review what we learned about in Activity 4, Gene Combo. In Gene Combo, we began looking at the first generation of critters, Sky and Poppy. Sky and Poppy were put in an experimental breeding program in which they then produced the second generation of offspring. In the second generation of offspring, all of the critters had blue tails, and it appeared as if the orange tail color trait disappeared. Two critters in the second generation, Ocean and Lucy, then were bred to create the third generation of critters. And the critters in the third generation had both orange and blue tail color. So it appeared as if the orange tail color actually did reappear. Okay. We also learned in that activity that blue tail color is dominant over orange tail color. Take it away, Mr. Rancourt.
All right, so I am on page 27 in the paperback book, okay? Activity 5, it's called Gene Squares.
So in our lab manual, it's going to be virtually page 38.
Okay, and I'm reading along and you're reading along with me. Activity 5, Gene Squares, problem solving. Joe wonders what chances he would have of transmitting Marfan syndrome to his children someday if he does have the gene. He asks the genetic counselor for help. She shows him a model to help him figure out the chances, but cautions him that the model predicts only what is likely to happen, not what will actually happen. In previous activities, you learned that each offspring receives half of its genes from one parent and half from the other parent. You also learned that different versions of genes are called alleles. You use coin tosses to model the way alleles are passed from parents to offspring. You observe that each offspring has two alleles, one from each parent. In the simulation to determine tail color, if one parent contributed a blue tail color allele, marked with the big T with the underline, and the other parent contributed an orange tail allele, the smaller t, the allele combination would be big T, little t. This is a critter's genotype for that characteristic. Because the allele for that blue tail color was dominant, the big T, little t combination caused the physical appearance of the tail to be blue. We call the critter's physical appearances its phenotype for the characteristic. So the genotype is the letter combination and the phenotype is the actual trait that you see that shows up. In this activity, you will learn how a table called a Punnett square can help you model and predict the genotypes and phenotypes of offspring.
So ultimately, the question that we're going to ask you to be able to answer at the end of this activity is this: how can we model and predict the ratios of traits observed in the offspring of parents with two versions of a trait? And this little diagram that you see at the bottom is actually the Punnett square that we're going to use to predict the genotypes and the phenotypes of the offspring.
Now we're good. Okay. So now I am on page 39 if you're using the online lab manual, or page 28 in the paperback. And we're looking at the material section. So the first thing you're going to need to do before we actually begin or the rest of this activity is either go into the DNA Diner and download Student Sheet 5.1 Punnett Squares, or the cool thing about LabAIDs right now is if you're following along in the book, you can actually click on this icon. It'll load it for you. Just make sure that you save it properly in Kami. You remember, download, save to your Google Drive with all annotations so that when you start marking on it, everything that you marked on it will actually save and hold into the system. Cool? So the procedure says this. So after you've got the Student Sheet 5.1 in front of you, the next thing you're going to do is you're going to read below how to use Punnett squares. Work with your group to prepare a short summary that describes how Punnett squares model the results of sexual reproduction. So as you're going through here and we're reading about how to create Punnett squares, keep in mind that eventually you're going to actually have to summarize this for us as well. Okay. So if you have any questions about how to do it, make sure you ask that before we start summarizing. And then step B says complete the Punnett squares on student sheet 5.1, Punnett squares step by step. And we'll have that. It's going to look like this document right there. Okay. So now Mrs. Shelton's going to read the next part, how to use Punnett squares background. And I'm going to slide this up.
All righty. So a Punnett square is a diagram you can use to show the likelihood of each outcome of a breeding experiment. It is used when each parent's genes for a trait are known. By filling in the squares, you can find the possible genotypes and phenotypes of the offspring. Remember that the genotypes are the 2-letter combinations for a trait, and the phenotype is the physical appearance that actually is shown by the trait. You can also predict the chances that each phenotype will occur in the offspring. Consider the cross between Sky and Poppy as an example. The 2 phenotypes for tail color are blue and orange. As you modeled with colored discs and creature features, there are 2 versions of the tail color gene, one for blue and one for orange. These 2 versions are called alleles. As you saw, when you did the coin tosses in Gene Combo, the blue allele can be represented as an uppercase T with an underline, and the orange allele as a lowercase t. The uppercase letter represents the dominant trait. You might also use a capital B for blue and a lowercase b for orange, since blue is the dominant trait. But you need to use the same letter, uppercase and lowercase, for the 2 alleles of any one gene. To avoid confusion, always remember to underline the uppercase letter. Because Sky is from an island where there are no orange-tailed critters, we can assume he has only blue tail color alleles. So his genotype for tail color is big T, big T. Because Poppy is from an island where there are no blue-tailed critters, we can assume she has only orange tail color alleles. So her genotype for tail color is little t, little t. An organism that has only one kind of allele for a characteristic is called homozygous. Let's remember that the prefix homo means same. Therefore, that if you have one kind of an allele, it's going to be homozygous, meaning the same. Sky is homozygous for the blue tail color trait, while Poppy is homozygous for the orange tail color trait.
Okay, so now if you're following along in the paper manual, in the paper book, it's to page 29, and I believe we're up to page 40 in the online version. So before we actually start this sheet right here, or this sheet right here, rather, we're going to tell you exactly how to fill one out, okay? So read along with us. So step 1 says this: every Punnett square should have a title. So notice the title here is tail colors, 4 boxes, the genotypes of each parent, right here, listed along the top, and the other parents' traits listed all along the left of the square, and a key. So you want to make sure that you have your title, make sure you have your 4 boxes, make sure you have both alleles, okay, or the genotypes here, all right, listed. And then you need to make sure that you state to the person who's reading your Punnett square what these letters actually mean. Remember, big T with the underline means blue allele because that's the stronger or the dominant trait. And in this exercise, the little t is going to stand for the orange allele because it is what we call like the weaker or the hidden trait.
Recessive trait.
Recessive trait. Thank you for correcting me. Write the possible alleles donated by each parent along the top of the table and the left side of the table. It doesn't matter which parent you use for each position. In the top left table shown above, Sky's alleles are placed along the top.
Remember, Sky was homozygous for blue tail trait. So he was big T, big T. So you take each of the T's and you separate them along the top of the Punnett square.
Got it. So it doesn't matter which parent you use for each position. In the top left table shown above, Sky's alleles are placed along the top and Poppy's are placed at the left. Each big T along the top represents an allele in the sperm cell produced by Sky, and each little t on the left represents an allele in the egg cell from Poppy. Step 2, completing a Punnett square. Complete each box of the table by combining one allele from the top and one allele from the left, as shown in the bottom table. So if you look right here, I'm going to take the big T right here, I'm going to transfer it down to here, and I'm going to take the little t from here and place it to the right of there. So I have big T, little t. And you're going to keep going. So when you combine one allele from each parent into a box, you are representing a sperm cell and a fertilizing egg. And you're going to do this for every box. So you'll notice right here, big T goes right here, little t goes right here. Big T will also go down to this box right here. Little t will go to that box right there. Remember, I have 4 squares, so I've got to complete the last one. So big T will carry down all the way to here, and the little t will carry all the way across from there. Step 3, making conclusions using a Punnett square. Now you can use the Punnett square to make some predictions. All of Sky and Poppy's offspring will have one allele for the blue tail color and one allele for the orange tail color. Notice that right here. Each box or each offspring has one big T, and each box has one little t. Their genotypes will be big T, little t. An organism that has alleles for 2 different tail colors is called heterozygous. So when you have a big letter and a little letter, that's heterozygous. If you have a big letter, that's homozygous, because remember, homozygous means the same. And also, if you have little t and little t, or 2 little letters, that also is homozygous, because still I have the same exact letters. It's only heterozygous when you have a capital letter and a lowercase letter. Okay. Because blue tail color is dominant over orange, the phenotype of all offspring is blue tails. Remember, phenotype is what you're going to actually see, what shows up, the trait. Okay? And since each one of these has a big T and a little t, you know that big T is dominant over little t, so we're getting blue tails. Out of every one of these offspring, they will all have blue tails.
Now let's remember that's just a possibility. It doesn't necessarily mean that's 100% going to be that way. Okay, but when we're looking at Punnett squares, it's really important that we understand that it's probable that this is what's going to happen.
Because blue tail color is dominant over orange, the phenotype of all offspring is blue tails, as found in the breeding experiment between Sky and Poppy. Hit it, Doors.
Hey guys, we're back. So what we want you to do now is we'd like you to pull up Student Sheet 5.1 that we asked you to access previously, and we're going to walk through with you the very first example of completing a Punnett square step by step. As I go through the directions, Mr. Rancourt is actually going to complete the very first example of a Punnett square with you. Please make sure, as he is modeling how to do this, that you also are following along and completing your student sheet, as you will be submitting this for credit. Okay, so what you see here is the cross between Generation 2 critters, Ocean and Lucy. Now remember, Ocean and Lucy are Generation 2, and their genotype is going to be big T, little t. So in other words, we've got Ocean being bred with Lucy. They are going to reproduce what are called the 3rd generation of critters. Their genotypes are big T, little t times big T, little t. Okay, we know that a big T represents the allele for blue tail color, and that blue tail color is dominant to orange tail color. Therefore, the little t is the allele for the orange tail color and is recessive. Okay, please note that while Ocean and Lucy both have blue tails, they are both heterozygous. Let's remember, the prefix hetero means different, which is why their genotypes are a big letter and a small letter combined, okay? So, step number 1 says, referring to the example above from your book, complete this Punnett square for the cross between Ocean and Lucy. Place Ocean and Lucy's alleles on the dotted lines in the Punnett square. So we're going to start with Ocean at the top, okay? And we're going to put Ocean's alleles at the top of the square. So it's going to be big T. Remember to underline that big T so you could see, and little t, okay? And then we're going to put Lucy's alleles on the left side, and Lucy is also big T, little t, which means heterozygous, okay? In step C, it tells us to use either a blue, we're on step B. Complete the Punnett square by filling in each box with the allele above it and the allele to its left. So this is how you actually go about completing a Punnett square. So we are going to take the allele from Ocean and cross it with the allele from Lucy in that very first square. In other words, we're going to take a big T and a big T, and we're going to combine them and put them in the first square. So this will be big T, okay? And in the second box, we're going to look at the alleles, and we're going to be crossing the little t with the big T. And the results are going to go into the second box. So you're going to end up with little t, big T. Now, guys, it really doesn't matter which order you put these in. I always prefer to have my capital letter first, but I think since you're new to learning this, it's okay if you just put them in the order in which you see them, because it doesn't matter. The outcome is going to be the same. For the third box, you're then going to cross Sky's big T with Poppy's little t. And when you do so, you'll end up with a big T, little t. And finally, the fourth box, we're looking at Sky's little t. I'm sorry, not Sky. Ocean's little t and Lucy's little t. And we're putting those in the fourth box. And so you're going to end up with little t, little t. Okay. Now, let's make sure we have our key complete. We know that big T represents the blue tail color trait. And we know that little t represents the orange tail color trait. So let's go ahead before we move on to steps C, D, and E, and let's talk about what the genetic crosses in each of these squares is going to yield. In the first box, we end up with big T, big T. So big T, anytime there's a capital T, we know that we're going to end up with a blue tail color trait, just like in activity 4 when you toss the coins. The combinations, the gene combinations are the same. In the second square, you have a little t, big T. That also is going to yield a blue tail. In the third box, you end up with a big T, little t, which is going to yield a blue tail. And in the fourth box, you have a little t, and we know that the only way that we can get this recessive trait to appear is if both of the alleles are actually lowercase. And so this particular square is going to yield an orange-tailed critter.
Can you explain one thing for me?
Sure.
So can you explain why a little t and big T little t are the same?
Well, they're exactly the same because it's the exact same combination regardless of which way you place the letters. And so anytime you have a big T, the big T is dominant over the little t, meaning it is going to overpower or overwhelm the little t.
Okay.
And so for that reason, this particular outcome is going to be a blue tail. Okay. And the same is true here. The big T overwhelms or dominates the little t. And for that reason, it's going to be a blue tail.
So it's not the order. It's just making sure it has to do with the actual letters that are placed in there.
That's correct, Mr. Rancourt.
I think that'll help some people a lot.
Good. Okay. So now we're going to use either a blue pencil or a regular pencil to shade in the squares for the offspring that will have blue tails. So let's go ahead and let's do that now. And you can see Mr. Rancourt is looking for his drawing tool with the blue. We're trying to find that pretty blue color. And we're going to color in the squares that are actually going to be blue. So we know the first one, big T, is in fact going to be blue. So you see him shading that in. We know that square 2, little t, big T, is also going to be blue. So he's going to shade that one in. Likewise, box 3 is going to be big T, little t, also blue. So let's move on to letter D. In letter D, it says about what fraction of the offspring of Ocean and Lucy are predicted to have blue tails according to the Punnett square. So if we look at the square, we realize that there are 3 of the 4 possible squares, or 3/4, that are going to be 3/4 are actually going to be blue tails. Okay, and then it asks in letter E about what fraction are predicted to have orange tails. And so we know little t is the only one that's going to yield an orange tail color trait. So only 1/4, okay, are predicted to have orange tails. Ladies and gentlemen, we hope that this example helps you to complete the remainder of Student Sheet 5.1. Please notice that if you scroll to the back of this page, there are 2 questions that you are responsible for. Just as Mr. Rancourt and I showed you, it is very important that you place the alleles at the top, okay, of each of the Punnett squares, that you cross them, completing the square itself, that you complete the key, and you shade it in and answer each of the questions. Again, you need to do this for both examples 2 and 3, and you will be submitting this for credit. Hope to see you guys soon. Hope you're well. Take care.